Hello, my dear students. Welcome you all to Sri Jaindra Rank Zone classes. I am your mentor and your friend for the subject biology, and I will be taking the zoology part, that is animal science. My dear students, in the last lecture, I have started the osmoregulation. So, starting from the topic of osmoregulation, I will be revising the points of the previous video, that what are osmoregulators, what is osmoregulation, what are osmoconformers. Okay. So osmoregulation is basically, or we will talk about the osmoregulators. Osmoregulators, these are the animals that can maintain a constant osmolarity of body fluids irrespective of the ambient environment. Ambient or external medium. Vertebrates, except myxine or hagfish. Myxine is a vertebrate, but all vertebrates are osmoregulators. Myxine is a osmoconformer. Okay. Osmoconformers, animals that change their body osmolarity, animals that change their body osmolarity of body fluids according to the ambient environment. According to the external medium. So invertebrates are osmoconformers. They are osmoconformers. Okay. There is a graph which is given organism in population between internal medium and external medium. The constant line is osmoregulator. Ones which constantly change their body osmolarity are osmoconformers, and the other categories partial osmoconformers. Partial osmoconformers, the most probable examples are elasmobranchs, cartilaginous fishes like sharks and torpedoes. Rays. They are partial osmoconformers. Okay. Now, seabirds, they excrete ions with the help of nasal glands. They are termed orbital glands of crocodile and turtles. Near eye excrete excessive salts. Okay. Now, osmoregulation in marine fishes. Sorry, osmoregulation in marine habitat. If the external medium is hypertonic, to body fluids. Okay, so marine habitat means the external medium is hypertonic to in respect of body fluids. Okay, the different problems which are faced. Body loses excessive amount of water, and body gains excessive ions. Elasmo branchs or cartilaginous fish and Latimeria coelacanth. Okay, they are the osmoregulators in marine habitats. They retain urea and TMAO, trimethylamine oxide, in blood so that the osmolarity of blood is raised. As a result, there is no loss of water. No loss of water, right? So elasmobranchs of cartilaginous fishes and Latimeria coelacanth, they retain urea and trimethylamine oxide in body fluids, okay, to maintain the to increase the osmolarity of blood so that body should not lose the excessive amount of water. Okay. Rectal glands actively excretes ions in marine habitats. Bony fishes or telotes, telos is a bony fish, okay. They also help to maintain the body osmolarity, especially in the marine habitat. Divalent ions like magnesium, sulfate are removed along with feces. And monovalentines like Na plus, Cl minus are removed actively by chloride cells or ionocytes. Okay, so divalent ions they are removed along with feces, and monovalent ions like Na plus, Cl minus are removed actively by ionocytes. So we have discussed the osmoregulation in marine fishes. We have discussed the osmoregulation in freshwater habitat previously. Okay. Now we are going to discuss the osmoregulation in terrestrial habitat. So I have to add a slide. Osmoregulation, terrestrial habitat. So we are going to discuss about amniotes having amniotic membranes like reptiles, birds and mammals, and insects. Insects they have a waxy coating epicuticle over exoskeleton, which prevents water loss. Which prevents water loss. Excretion of uric acid, which requires least amount of water, also help to maintain the water balance. Okay. Amniotes they have keratinized stratum corneum, that is skin, which prevents water loss. Kangaroo rat is a desert mammal, only mammal which never drinks water. As telosts, bony fish, they don't drink water. and they excrete dilute urine. Drinks water. Kangaroo rat. Nocturnal animals remain in their burrows during daytime to avoid body perspiration, to conserve water balance. They have nasal countercurrent heat exchange mechanism. Counter current heat exchange mechanism. Okay, so because of this nasal countercurrent heat exchange mechanism, okay, they can retrieve the water vapor from the air to be exhaled. They can absorb the water vapor from the air to be exhaled. They retrieve the water vapor from the air to be exhaled. They produce highly concentrated urine. They utilize metabolic water, which is produced as a result of oxidation of food, to maintain the water balance. Okay, so this is for kangaroo rat. Now, camel, it is also a desert mammal. Water is stored in the tissues and then they can survive without water for many days. They also excrete highly concentrated urine. They store fat, food in hump. Their bodies show high tolerance for increase in temperature, xerothermals. Okay, humans cannot tolerate if the water loss is 12%, but camels can tolerate dehydration of twice of this value. but camels can tolerate dehydration of twice of this value. So, ladies and gentlemen, we have completed the osmoregulation, osmoconformation part. Okay. In the next video, I will be starting with the artificial kidney. Okay. Till then, bye bye. Take care. Stay safe. Stay healthy.